Yeah. It's one of those days. So, we have quite a bit of content to get through, so we're going to get through it. So, respiratory limitations to exercise tolerance. I really like this slide. A runner's view of physical training. Okay? So the general idea is that you have a healthy untrained individual on the left and an aerobically trained individual on the right. So what you're looking at here is basically kind of like the lungs, the chest wall, the heart, and then the peripheral locomotor muscles in the healthy untrained individual. So the idea here is that you have a circumstance or a physiological system where the lungs and the chest wall are so overbuilt that they can accommodate the demands of the heart and on the peripheral muscles. So the idea here is that in a healthy untrained individual, the primary determinant, the thing that lags behind and the thing that contributes to exercise intolerance is basically aspects related to the peripheral muscles and the capacity of the heart to deliver oxygen to them. The respiratory system is relatively overbuilt and can outrun the capacity of those to the muscular and the cardiovascular support systems. But in an aerobically trained individual, somewhat paradoxically, is that there are the heart and the peripheral muscles, so there are adaptations of the periphery and centrally that occur to actually make the muscles that much more energy efficient in terms of increasing mitochondrial content, enzyme capacity, capillarization, increases in muscle mass, density, various other factors. And so, in contrast to what happens to the peripheral muscles and cardiac structure and function is that the respiratory system does not adapt to training. So there are not increases in the surface area for gas exchange. There's not dramatic increases, if any at all, in terms of respiratory muscle strength. There's no changes in airway diameter. There's no changes in airway length that we know of with aerobic exercise training. And so the idea here is that we have a circumstance where in healthy untrained individuals is that the respiratory system is overbuilt for the demands that are imposed on it by the cardiovascular system and the peripheral muscles. Whereas if you train, you actually can get a muscular system and a cardiovascular system that can outstrip the capacity of the lungs and the chest wall, is generally the idea. So in aerobically trained adults, the respiratory system at the very least has the potential to limit exercise tolerance. It's generally not a contributing factor in relatively in recreationally active and non-elite trained individuals or endurance trained individuals, but it can become a factor the more fit that you get, because the respiratory system doesn't adapt to the exercise training stimulus. So if we consider a couple of these more likely potential respiratory limitations to exercise tolerance in trained athletes, one is exercise-induced arterial hypoxemia, and two of the proposed mechanisms are a ventilation perfusion mismatch and a pulmonary capillary diffusion disequilibrium. Basically, cardiac output is so high that the red blood cell transit time is so short, the red blood cells spend such little time in the pulmonary capillary that there's not enough time for it to fully equilibrate with oxygen. Okay? And then also some evidence that I'll show you about this concept of a respiratory muscle steal phenomenon, where we have a relatively fixed cardiac output, and so if the work of breathing elevates, so we know now that ventilation increases as a function of metabolism. If you can train your muscles to achieve incredibly high metabolic demands, ventilation needs to track that. If ventilation goes up, the work of breathing goes up. If the work of breathing goes up, the oxygen cost of breathing goes up. If the oxygen cost of breathing goes up, the amount of oxygenated blood and the percentage of cardiac output that needs to go to the respiratory muscles needs to go up. So now you can see that you have a competition between the available cardiac output that needs to go to the heart, to the brain, to the quadriceps, for example, during cycling exercise, but all of a sudden now you need a higher percentage of that cardiac output to go to the respiratory muscles. So if most of the blood is being redistributed to the limbs, and now all of a sudden you have this very high work of breathing, you almost have to shift some of that blood away from the limbs to be able to support the respiratory muscles. And I'll show you some evidence that in fact in healthy, in very trained athletes, this is exactly what happens. Okay? So, what is exercise-induced hypoxemia? The name implies that it's a reduction in blood oxygen levels in the context of exercise. So the definition is arterial oxygen desaturation of greater than 3–4% below resting levels during heavy or near maximal exercise. So why would we say that it would have to be greater than about 3 or 4%? Think about the oxyhemoglobin dissociation curve, and think about the effect of exercise-induced increases in temperature and changes in pH. Can somebody explain the Bohr effect to me? So the rightward and downward shift of the oxyhemoglobin dissociation curve will result in a natural or normal 3 to 4% reduction in oxygen saturation with no change in arterial PO2, right? So at any given PO2, oxygen saturation falls. So remember we said that the Bohr effect will account for about a 3 or 4% decrease in oxygen saturation from rest to heavy exercise. Anything that existed above and beyond that is hard to ascribe to exercise-induced changes in temperature and pH alone. So anything that falls outside of that 3 to 4% would be considered some type of abnormal exercise-induced desaturation. Okay? So the prevalence of this condition, of this abnormally desaturated blood, is about 25 to 50% of endurance-trained athletes with a VO2 max greater than 65 mils per kg per minute. So these are very trained. These are like elite rowers, cyclists, marathon runners type things, okay? So very high VO2 maxes or very VO2 peaks. And the characteristics of this exercise-induced hypoxemia is that you have an abnormally decreased arterial partial pressure of oxygen level and an abnormal widening of the alveolar to arterial oxygen difference, with arterial oxygen desaturation, again, of greater than 3 to 4% below resting levels. And ultimately, this reflects incomplete oxygenation of the mixed venous blood in the pulmonary capillaries. So there's just not enough reoxygenation of that very hypoxic blood that's returning from the exercising tissues. Okay, so either it's not being ventilated enough, the lung is not being ventilated enough, perfused enough, or the red blood cell is not spending enough time in a well-ventilated and well-perfused lung. Okay? So what you can appreciate here is that if you look at rest, we have oxygen saturation, the arterial PO2, the alveolar to arterial oxygen difference, and the partial pressure of carbon dioxide in the arterial blood. And in healthy trained and untrained individuals at rest, we got a saturation of about 97%, an arterial PO2 of about 92, AADO oxygen difference of about 10, and an arterial CO2 level of about 40. In an untrained individual, you can see that at VO2 max, saturation levels will drop by about 2 or 3%, which we know is normal. Arterial PO2 levels are going to be maintained well preserved, so the ventilatory system is able to support the maintenance of oxygenation of the arterial blood. The AA gradient will widen a little bit, but not dramatically. There is some ventilation perfusion mismatch that does occur with exercise, and it actually, you can see a pretty substantial blowing off of carbon dioxide relative to rest, which we know now is a form of respiratory compensation for the metabolic acidosis. We try to blow off or increase ventilation disproportionately more than carbon dioxide production during heavy exercise to get rid of carbon dioxide, which in and of itself is a volatile acid and can liberate hydrogen ion. So if we get rid of it, we have less of an acid load, which counteracts the metabolic acidosis that's being produced by those exercising muscles. But in a trained individual, you can see that oxygen saturation levels can drop relative to rest by as much as 10, and in very extreme cases, even more than that percent. You can see that the arterial PO2 levels can drop down into, you know, the 70 millimeter of mercury range. The alveolar to oxygen gradient widens, which means that there's not a nice equilibration or matching between basically the alveolar PO2 and the arterial PO2, so there's a disequilibrium between oxygen exchange between the lungs and the blood. And you can see actually that while there is still a hyperventilation relative to rest, compared to the untrained individuals, there's actually some carbon dioxide retention. So you can imagine here that these individuals are hypoventilating in some ways. They're not ventilating enough to blow off the carbon dioxide levels, at least not to the same as the untrained individuals. And keep in mind that this is occurring in the setting of much higher metabolic demands. So we're talking 40 mils per kg per minute at peak versus 65 mils per kg per minute at peak, which means that we're also talking about much higher rates of carbon dioxide production, right? Because we know that VO2 and VCO2 are intimately related. If VO2 is higher, VCO2 is certainly going to be higher as well. So you can see that there are clearly some gas exchange disturbances, desaturation of the blood, failure to maintain arterial PO2 levels, as well as some carbon dioxide retention reflecting some relative alveolar hypoventilation. And so one of the mechanisms of exercise-induced arterial hypoxemia is this concept of ventilation-perfusion mismatch, which is really a relationship between alveolar ventilation and pulmonary blood flow, which is ultimately determined by cardiac output, okay, and local vascular resistances in the pulmonary capillaries. So if you look here, right, in this circumstance where we have a circumstance in lung A, where we have no impairment to ventilation and no compromisation to perfusion. So here we have a situation where oxygen inflow is equal to outflow, and so we have this perfectly ventilated lung. This is where ventilation and perfusion are perfectly matched. There's no compromisation on either end in terms of ventilation or perfusion, and so we have a nice harmonious exchange of oxygen from the lungs to the blood. In B, we have a situation where inflow, which would be alveolar ventilation, is less than outflow, which would be pulmonary perfusion, okay, or cardiac output. So you can see that here there's a block to pulmonary ventilation. So in other words, we're not delivering any fresh gas to the lung, but nevertheless, blood is being cleared out of the lung. So under these circumstances here, if we're not delivering oxygen into the alveolar space, but oxygen is exchanging and moving into the mixed venous blood, you're going to have a part of the alveoli that's going to have a very, very low alveolar PO2. So within that alveolar unit, there's basically going to be no oxygen, which means that there's no pressure gradient for oxygen exchange from the lung to the blood at that alveolar unit. Okay, we're talking about like an alveoli connected to a pulmonary capillary, but multiplied over millions of individual units, obviously. And if you look at C, here's a circumstance with a high ventilation perfusion ratio, where here we have no obstruction to alveolar ventilation, so basically no mechanical derangement, no inability to get air, fresh gas to the lung, but we have a part of the lung that's not being adequately perfused. So under these circumstances, we actually have a very high pressure gradient for oxygen exchange, which in this case would actually serve to maintain oxygen saturation levels, because we would have a situation where whatever blood is getting through, the red blood cell is moving very, very slowly, and there's a very high pressure gradient for oxygen exchange from the lung to the blood. So in this case, we have basically a perfectly ventilated and perfused lung in areas of low ventilation perfusion and high ventilation perfusion. So if we accept that the lung is going to have areas that are perfectly ventilated and perfused, areas that have low VQ and high VQ mismatched, okay, is that if you actually look in areas where, let's say for example, you have a low ventilation perfusion relationship where the alveolar ventilation is 1 relative to a cardiac output of 10. You can see here that the oxygen concentration, as this blood moves through the capillary, pulmonary capillary, oxygen saturation of the mixed venous blood is about 14.6 milliliters of oxygen per 100 mLs of blood, and then it rises as it leaves that pulmonary capillary to 16. In a perfectly ventilated lung, you can see here that it rises to about 20, and in an area with high VQ, you can see that it rises again to 20, where we have very nice reoxygenation of the blood. And ultimately, all of these alveolar units will combine, right? The blood from this, the oxygenation status from this alveolar unit and that alveolar unit and that alveolar unit will combine to mix to represent the total arterial oxygen content. So we have blood that's coming from high and low and perfectly ventilated alveolar units that are contributing to the total arterial oxygen content. So it's like the whole is the sum of its parts, okay? So if you look on the oxygen saturation curve, where we have oxygen saturation relative to the partial pressure of oxygen, and we have the arterial oxygen content here on the second y-axis, you can appreciate that areas of low ventilation perfusion mismatch or ventilation perfusion ratios compromise pulmonary gas exchange and arterial blood oxygenation. So you can see here that basically these areas would fall at this arterial oxygen content at a low PO2, and therefore that part of blood leaving that alveolar unit would be quite desaturated, have a low PO2 and a low saturation. Whereas areas with high or high ventilation perfusion ratios actually fall up along this upper alinear portion of the oxygen dissociation curve, where you can tolerate differences in the PO2 without much in the way of a change in oxygen saturation. So areas of high ventilation perfusion ratios facilitate pulmonary gas exchange and arterial blood oxygenation. But again, because you have all of these different parts of the lung that are contributing to the arterial blood CO2 and arterial blood oxygen content and saturation levels, you can appreciate that the effect of low ventilation perfusion ratios on arterial blood oxygenation is actually greater than equal or opposite effect of high. So in other words, because many of these low VQ ratios are operating on this very, very steep part of this oxygen dissociation curve, a given unit change in PO2 or arterial oxygen content coming from these low VQ units actually has a much more detrimental effect per unit change in oxygen content or per unit change in PO2 on oxygen saturation than the blood coming from the high VQ units. So the idea then is that if you had a similar number of high VQ units and low VQ units, the detrimental effect of the low VQ units is much greater than those on the high, because they result in much more desaturation of the blood. So ventilation perfusion mismatching actually tends to increase a little bit with increasing exercise intensity and is the primary contributor to the development of exercise-induced arterial hypoxemia in athletes. And this is because they can generate relatively high cardiac outputs, but because the respiratory system doesn't adapt to exercise training, they are not able to generate higher alveolar ventilations. So ironically, they push themselves into a situation where they create an inflow and outflow imbalance. They can generate such high cardiac outputs without much in the way of improvements in alveolar ventilation. So these trained athletes actually will end up having more ventilation perfusion mismatch because more blood is moving through the lung where there's not much in an adaptation to increase alveolar ventilation to try to accommodate or match those very high metabolic demands. So we have more of these units operating on this very steep and detrimental part of the dissociation curve than these high ventilation perfusion units. And again, that's because the adaptations to stroke volume and cardiac output are disproportionately greater in a trained individual than they are to the ability to increase alveolar ventilation. And so the trained athlete has more of an imbalance between oxygen inflow to the lung and outflow from it. So ventilation perfusion mismatch is one of the primary, if not the primary determinant of exercise-induced hypoxemia. Another potential mechanism is pulmonary capillary diffusion disequilibrium. So the preamble here is that during heavy exercise in elite athletes, again, VO2 is greater than about 65 mils per kg per minute. Pulmonary blood flow, which is oxygen outflow from the lungs, is very high due to the very high maximum cardiac output. Consequently, the amount of time that the red blood cell spends in the pulmonary capillaries may actually be less than the time required to complete oxygen equilibration between the lungs and the mixed venous blood. As a result, the alveolar to arterial oxygen difference widens, while oxygen saturation and arterial oxygen content decreases. So let's say, for example, here that we have the time of the red blood cell in the pulmonary capillary, starting from its appearance to when it leaves the pulmonary capillary. So let's say it's in there for 1 second. Okay? We have the partial pressure of oxygen. Here at rest, the alveolar PO2 is about 100 millimeters of mercury. Okay? So the mixed V, so during near maximal exercise, we know, and we've shown this before, that the alveolar PO2, because of an increase in alveolar ventilation, will rise. So we increase that high energy component for oxygen exchange from the lungs to the blood. So we widen that alveolar to mixed venous PO2 gradient. Not only by having progressive reductions in the partial pressure of oxygen in the mixed venous blood returning to the lung, but also by increasing the alveolar PO2. So we decrease the low energy component of the gradient, and we increase the high energy component to widen the pressure gradient in total. So we know that the alveolar PO2 widens. So what happens is that the mixed venous PO2 comes in at a very, very low partial pressure, let's say of about 15 to 20 millimeters of mercury. And as that red blood cell traverses through that pulmonary capillary in the setting of the elevated alveolar PO2, what happens is that you can see that it reoxygenates itself, and eventually, after about 0.3 to 0.4 seconds, you get complete equilibration between the red blood cell and the lung. So now that individual red blood cell has spent about a third of a second in the pulmonary capillary, and that is enough time at that pressure gradient to reload that red blood cell to be 97% saturated, for example. Okay? So the red blood cell transit time is equal to pulmonary capillary blood volume, so the total amount of blood in the pulmonary capillaries divided by pulmonary blood flow. So in an untrained individual, where the maximum cardiac output is 20 liters a minute or 355 milliliters per second, and generally it's considered that the lung gets all the blood all the time. Okay, so the idea here is that we have a pulmonary blood flow of about 335 mLs per second, and let's assume that we have a capillary pulmonary blood volume that's not going to change with training status of 210 mLs. So under these circumstances, the red blood cell transit time would be about 0.63 seconds, which is twice as long as required for that red blood cell to fully equilibrate in the pulmonary capillary. It needs about 0.3 seconds, but the time it spends is about 0.63, based on how much blood is in the pulmonary capillary and how quickly it's moving through the pulmonary capillaries. Now, so you can see that's the time required. This is the time required, and this is the time that it spends. Now, let's take an endurance-trained athlete with a peak VO2 of, say, 65 mils per kg per minute. Now, they have almost a twofold increase in cardiac output at the same pulmonary capillary blood volume. You can see now that the red blood cell transit time is really flirting with that minimum amount of time that's required to spend in the pulmonary capillary. Now it's down to about 0.3 to 0.4 seconds. So in this case, you're flirting with a circumstance where the red blood cell is just not spending enough time in the pulmonary capillary before it exits to actually create this complete oxygen equilibration. And so you might have red blood cells that are leaving that are not fully saturated. And so all of those red blood cells together are going to contribute to a desaturation of the arterial blood that's going to leave and be sent towards the tissues. Now, you could facilitate this exchange even more. You can offset the adverse effects, potential adverse effects of a very short red blood cell transit time by further increasing the alveolar PO2. So if you could elicit a greater alveolar hyperventilation response and increase alveolar PO2 to 140 millimeters of mercury, you would get a greater driving pressure and probably shorten the time required that the red blood cell would need to spend in the pulmonary capillary. But if the respiratory system does not adapt to exercise, you can't generate higher alveolar ventilations, which means that you can't bring in more fresh gas into the lung, which means that you have a limit to which you're able to increase your alveolar PO2 to try to offset the shortened amount of time that the red blood cell would spend in the pulmonary capillary. So there could be literally a mechanical ventilatory limitation to exercise, where you want to breathe, there's a metabolic stimulus to breathe, but because the respiratory system doesn't adapt, the lungs don't get bigger, the respiratory muscles don't get stronger and more endurant, to, or resistant to fatigue. You don't generate more alveoli. You don't get, more airways. Is that the net effect is that you kind of have a limit to the extent to which you can increase your alveolar PO2 and the driving pressure for oxygen exchange from the lungs to the blood. So we have two important mechanisms that can contribute to that exercise-induced arterial hypoxemia in athletes. But what are the consequences of this exercise-induced hypoxemia? We know it occurs in 25 to 50% of athletes. We have some idea of why it occurs, but does it really impact on exercise performance? If it didn't impact on exercise performance, why would we really care? But we have a pretty strong appreciation now from the Hogan and Richardson studies, those calf plantar flexion exercise studies, and also the Hazeler studies where they did the oxygen manipulations and you saw the 31 pNMRS measures of phosphocreatine and inorganic phosphate changing with the inspired and expired levels of oxygen during both graded and constant load exercise. Right? We showed some of those magnetic stimulation studies and the time trial studies of Marcus Amen, where we showed that exercise time trials vary in relation to inspired oxygen levels. So even relatively small changes in oxygen status of the muscle could shift the balance between sustainable sources of energy provision and not. Then you're getting into the net drive and O2 conformer hypotheses and the integrative physiology that this class is trying to establish. So what are the consequences of exercise-induced hypoxemia? So this is work by Lee Romer, who's now a professor in, I believe, University of Exeter. He's a prof in the UK. He trained with Jerry Dempsey at Wisconsin, so this is one of another Jerry Dempsey product. This is actually from Jerry Dempsey's lab. And so this is 11 male endurance cyclists, about 60 mils per kg per minute in terms of their peak VO2. And in this study, subjects exercise on the cycle ergometer at a very high intensity, so near maximal intensity to exhaustion, during which time arterial oxygen saturation fell from 98% just down to 92%. So this was exercise-induced arterial hypoxemia, but it wasn't very severe, but it was nevertheless greater than could be explained by the Bohr effect. So about a 6% drop in oxygen saturation from rest. So then, on a separate day, separate occasion, the participants repeated the exercise, and again, this is very key, for the exact same duration and at the exact same power output that they did under the control condition, but breathed gas mixtures that literally prevented exercise-induced hypoxemia. They didn't give them tons of oxygen, they just gave them enough to prevent that 6% fall. Because what the researchers wanted to do was, how important is that 6% fall? I don't want to give them 60% oxygen if all I want to know is what is the effect of that 6%. So they just gave enough to prevent the 6%. It's a really nice kind of physiological study because you can overkill something by giving 100% oxygen, but in reality, you really just want to know what that 6% does to performance in these trained cyclists. And they looked at quadriceps fatigue assessed using stimulation of the femoral nerve. Again, on a chair, on a bed, leg strapped into a device that doesn't let it move, and they stick an electrode into the femoral nerve and they shock it, the leg tries to kick, but it doesn't move, and it measures the strength of contraction or concentric contraction against basically a force transducer, or a pressure transducer. And the pre to post exercise loss of force production to a maximal nerve stimulation is an indication of muscle fatigue. You innervate that muscle with a maximum stimulation. If it produces less force, the muscle's fatigued, right? So that we've seen that in a couple of slides before. So this should be familiar. This is the study design. They start, in this case, they exercised at 90% peak VO2 was equivalent to 294 watts. And to put that, I mean, they exercised at 294 watts for 13.2 minutes in the control condition when they experienced a 6% desaturation, and they brought them back in. They gave them a little bit of oxygen to prevent it, but then they stopped them at 13.2. So again, this is a ISO time measure. You don't know what's happening to the muscle if you don't stop them at the same time. Because if you stop them at peak and this group was able to go on longer, presumably they generate the same amount of fatigue. I'll encourage you to go back and look at those studies where they did constant load exercise with the different oxygen levels as well as the time trial. Remember the time trial study? Normoxia, hyperoxia allowed them to complete 5 kilometers faster. Hypoxia allowed them to complete 5 kilometers shorter, but when they twitched the muscle, the level of fatigue development was the same despite differences in performance. So again, you need to standardize the time point to know what's happening, get a good look inside the muscle, what's happening in the muscle. So, pretty remarkable effect, to be honest, from a physiological perspective. If you look at the magnitude of quadriceps twitch force as the percentage change from pre-exercise rest, remember, the more negative it gets, the more fatigued the muscle. The greater loss of force the leg was able to generate to a maximum stimulation of the nerve. And when they prevented, they did 13.2 minutes of exercise at an average of 294 watts in the setting of hypoxemia and just preventing it by titrating in a little bit more of inspired oxygen. They didn't completely eliminate peripheral muscle fatigue, but they attenuated it or prevented its development after that period of time by a pretty substantial amount. They halved it. So you can see that now we're about a 15% loss versus about a 35% loss. So that 6% desaturation, that difference that they presented, had a dramatic effect on exercise performance and the rate of development of peripheral muscle fatigue. So the idea here is that impairments in oxygen exchange that seem to occur, exercise-induced hypoxemia that occur in a quarter to half of elite athletes, through those mechanisms that I showed, have a true effect downstream on the muscle, presumably because it's resulting in a compromisation of oxygen delivery. Remember, at a given amount of leg blood flow, a reduction in arterial oxygen content will compromise intracellular PO2. And you should be able to work through all of the compensatory mechanisms and fatigue-causing mechanisms that would explain this. So the oxygen desaturation that normally occurs in endurance-trained subjects during high-intensity exercise while breathing room air. So again, just like going out and doing a heavy-intensity exercise in the gym or in the street, contributes significantly to the development of quadriceps muscle fatigue and therefore maximal exercise performance. So, that 6% is meaningful. It has an impact on human performance because it accelerates the rate of muscle fatigue development. So, some studies, they've pooled data from multiple exercise-induced arterial hypoxemia studies to try to get a sense of what is the threshold and impact on VO2 max for reductions in oxygen desaturation. And basically the threshold for exercise-induced hypoxemia in terms of creating a performance limitation occurs at oxygen desaturation levels greater than 4%. So again, we know now that about a 4% drop in oxygen saturation is normal and can be explained by temperature and pH changes through the Bohr effect. Anything above and beyond that is considered abnormal and actually begins to have an impact on oxygen desaturation. So the idea is that beyond about a 4% drop of saturation below resting levels, each further 1% decrease in oxygen saturation results in a 1 to 2% decrement in peak oxygen capacity, or VO2 max. So again, that's consistent with what I showed you before. Relatively modest changes in PO2 when you prevent them have pretty dramatic impact on the rate of development of peripheral muscle fatigue development. So if you're able to achieve higher power outputs, you can achieve higher VO2s. And you can see that as the blood becomes increasingly more desaturated, is that you get progressively greater falls in peak exercise capacity. So this exercise-induced hypoxemia does have consequences on exercise performance. So if we consider the other potential mechanism of a respiratory limitation exercise tolerance, which would be this respiratory muscle steal phenomenon, there are physiological consequences of an exaggerated work of breathing. Okay, so again, if you're able to achieve very high metabolic demands, that puts a very high demand on your respiratory system, which hasn't necessarily adapted to chronic exercise training. Higher ventilatory demands means higher work of breathing. Higher work of breathing means higher oxygen cost of breathing. Higher oxygen cost of breathing means more requirement for delivery of oxygen and blood flow. So the respiratory muscles are actually well designed to compete for a significant percentage of the available cardiac output during high intensity exercise in both health and disease. We know that the respiratory muscles demand a significant amount of blood during dynamic exercise, so it's about 8 to 10% of maximum cardiac output in untrained adults, and it can rise to about 15 to 16%, at least on the experimental estimates, of maximum cardiac output in endurance-trained adults. So the respiratory muscles can demand a higher percentage of the available cardiac output. So again, there's this potential for stealing blood away from the quadriceps when they require more blood of the available cardiac output. The respiratory muscles are innervated with nerves that are sensitive to metabolites, so they have these metabosensitive receptors. Okay, so like I showed you with peripheral muscles, if the respiratory muscles kind of get fatigued and metabolically perturbed, they can be stimulated and send sensory afferent information to cardiovascular and respiratory control centers to, say, signal an increase in ventilation, but also an increase in muscle sympathetic nerve activity, which we know would send a vasoconstrictor outflow to the peripheral locomotor muscles, including those that are exercising. That has the potential to compromise limb blood flow. The interesting thing is that the diaphragm, for example, the major muscle of inspiration, is not heavily innervated in terms, does not have many alpha-1 and alpha-2 adrenergic receptors. So the idea is that sympathetic activation, so if you activate the sympathetic nervous system, a given amount of sympathetic nervous system activation will preferentially affect the limb muscles and will not necessarily affect the diaphragm, because the diaphragm is not susceptible to vasoconstriction, at least not as much as the peripheral muscles. So the respiratory muscles are less susceptible to sympathetically mediated constriction of blood flow. So again, you have this respiratory system that can signal an increase in muscle sympathetic nervous system, but is resistant to the constricting effects of it. So again, you can already see that there's these mechanisms in place that would maybe allow for a relative distribution of blood away from the peripheral muscles towards the respiratory muscles. And during prolonged and heavy intensity exercise, respiratory muscles can activate the sympathetic nervous system, as I mentioned, compromise exercising limb vascular conductance and therefore blood flow. This would have the effect of decreasing oxygen transport to the exercising muscles, compromising intracellular PO2, requiring an increase in glycolytic flux and phosphocreatine degradation to support the ATP demands, increase the rate of development of fatigue causing metabolites, increase intracellular inorganic phosphate levels, hydrogen ion levels, compromise metabolic and contractile function, and that's the fundamental component of another form of respiratory limitation exercise. When the respiratory muscles demand more blood, it just takes more away from the limbs. So, start providing you some experimental evidence of this. So, this is really a summary of what I showed you before, but here you're looking at the work of breathing relative to minute ventilation and respiratory muscle VO2. So let's say that you go from rest and you increase ventilation. A 30-year-old sedentary male with a VO2 of 35 to 40 mils per kg per minute can achieve a peak ventilation of, say, 125 liters a minute, requiring a work and oxygen cost of breathing of about 250 joules per minute and about 350 mLs of oxygen per minute. Now, because the metabolic demands of a trained athlete are so much higher, they're able to drive their ventilation to much higher levels and actually get this disproportionate increase where you can see there's a doubling of the work and the oxygen cost of breathing. And so if you look at the proportion of the cardiac output in a trained individual, about 75 to 80% of the cardiac output is going to the limbs, about 15 to 20% is going to the respiratory muscles, and the rest of the blood is going to other places, so heart, brain, non-exercising tissues, okay? Liver, gut, kidney, things like that. So the work and oxygen cost of breathing at different levels of ventilation during exercise in a sedentary and endurance-trained individual are presented here in the graph. And at maximum exercise in the athlete, respiratory muscle VO2 approaches 15 to 20%, 16% in this case of whole body VO2, and presumably therefore the respiratory muscles require a similar proportion of the cardiac output, which is considerably more than that is required in the sedentary adult. Okay? Take-home message, the respiratory muscles, higher VO2, higher blood flow requirement, higher oxygen delivery requirement. So if you looked at the relationship then, so there's been a series of studies that I'll show you that have manipulated the work of breathing. They've either experimentally increased it or decreased it, and looked at the consequences of rate of development of muscle fatigue, muscle blood flow, and various other factors. Okay, so there's some nice research that I'll show you. But here you're looking at blood flow to the legs and to basically everything else relative to cardiac output. So here you've got a cardiac output that's about 30 liters a minute. And here you have a circumstance where you're exercising an individual with a normal work of breathing. No imposition into the respiratory circuit at all. So you're just increasing your work of breathing to whatever it is, 600 joules per minute, for example. Now, if you load the respiratory system, let's say that you make the inspiratory muscles work harder by putting a resistor into the inspiratory line. So let's say that you take like filter paper, and you put filter paper on every breath. You have to basically breathe harder to generate flow. So remember, flow is the relationship between the pressure gradient and the conductance. So if you decrease the conductance, you have to maintain a given flow, you have to generate more pressure. The respiratory system to generate more pressure means more forceful contraction. More forceful contraction means higher oxygen demand because more metabolic requirements. So you can impose a load on the respiratory system by adding a resistance on inspiration. And so you can increase the work of breathing experimentally, and when they do this, it had no effect on cardiac output, but actually they found that blood flow to the legs decreased in the high work of breathing condition. And presumably the increase in blood flow to everything else was a redistribution of blood away from the limbs to the respiratory muscles, because the idea was that everything else during exercise was kept the same, other than the experimental increase in the work of breathing. So this was kind of the first observation that was done, that perhaps there can be this stealing of blood during high work of breathing conditions away from the lower limbs or the exercising limbs toward the respiratory muscles. There can be this redistribution of blood flow. So Craig Harms, again working with Jerry Dempsey, about 20 years ago now, did a pretty important study where they looked at the relationship between whole body VO2 and exercise time during constant load cycle exercise performed at 90% of VO2 max, so a very high exercise intensity. And they did a condition where they looked at a control condition, so there was no intervention on respiration or the work of breathing at all, and then they unloaded the respiratory muscles. Because the loading experiments are great to say that if I bring you into the lab and I load you up, so I make anything harder, so like I do dead space loading or body mass loading, for example, I would just know that in the setting of a load or a stress, you could steal blood away from the respiratory muscles, from the peripheral muscles to shift them towards the respiratory. But I don't know that the normally occurring load on the respiratory system that occurs during normal exercise is actually contributing to a shift in the blood distribution, right? So what I would want to do is I would want to unload the work of breathing, and if I got improvements in exercise time and improvements in limb blood flow, I would know that the normally occurring work of breathing is having a negative impact on muscle blood flow. Because you can load something just means that it's possible. If you unload the system, that means that in the setting of normal exercise, it's actually having an adverse effect. So the unloading experiments are sometimes more challenging, but they're more physiologically relevant. So here they unloaded the respiratory system, and what they found was that they got a 15% improvement in exercise time without much in the way of differences in whole body VO2. So respiratory muscle unloading. So basically they put these subjects and they make them breathe on a mechanical ventilator, where the ventilator will do much of the work by pushing pressure into and out of the lungs and unloading the respiratory muscles. So the breathing stays the same. You can generate the same ventilations. It's just that you have a machine that pumps, that creates some of the pressure gradients in the respiratory system to unload the work that the respiratory muscles have to do. And it improved exercise endurance time by 15%, indicating here, just descriptively, that the work of breathing has the potential to compromise exercise performance, but it didn't necessarily provide the mechanism as to why. So, the work of breathing normally incurred during sustained heavy exercise intensity has a significant negative influence on exercise performance, because when you took it away, performance improved. So, what are some of the candidate mechanisms or pathways for this? Here we have the relationship between the respiratory musculature sending afferent, sensory afferent information to the brain that can then signal sympathetic vasomotor outflow. So you can basically get this respiratory muscle mediated sympathetic activation. So the respiratory muscles become overburdened and start to fatigue. You get reflex activation of these muscle metabolites, so these metaboreceptors send information to the central nervous system to reflexically increase sympathetic vasomotor outflow. That will then act on alpha-1 and alpha-2 adrenergic receptors in the periphery, will cause vasoconstriction. So you get an increase in sympathetic efferent discharge causing limb vasoconstriction. We know now that that has the potential to compromise oxygen output and increase the rate of locomotor muscle fatigue development. So this is kind of the concept of respiratory muscle steel. Load the muscle, activate the sympathetic nervous system, and has a preferentially greater effect on the limb. If the limb constricts without changing cardiac output, the blood is probably going to be redistributed to those loaded respiratory muscles. So, other work that was done by Craig Harms with Jerry Dempsey looked at experimental manipulations of the work of breathing during exercise, where they loaded and then unloaded the work of breathing and did some really nice measurements of